Is the skull information that will be in our exam? The position of the teeth in the human skull will be something that you need to know. So you need to know, for example, that the incisors are the teeth at the front, the canines are the next one along, and then the premolars, and then the molar. Do you remember when we were in class and we showed each other our teeth and we took each other through whichever teeth were which? Yeah, basically that. So the answer is yes. Okay, so know that your molars are right at the back, and know that your molars have these features. Remember when I went through the past paper question and showed you exactly what they wanted you to highlight for teeth? Okay, so make sure that you know what the features are for each of the teeth, and make sure that you know what the functions are for each teeth. Make sure that you know all the different stages for tooth decay. Make sure you can give a step-by-step method of tooth decay, if you want to think of it like that. Okay, the action of the plaque and the acid and gum disease. Okay, so we practiced that. Okay, make sure that that's on this section here. Boomy, do we have to study the diagram? Yeah, that diagram? Yes, absolutely. You need to know all the little bits. You need to know about the fibers attaching tooth to jawbone. You need to know about the pulp active cavity. Everything that is here is endorsed by Cambridge. So literally, if you learn that book, okay, you learn everything by heart, all the features, everything like that, you'll get an A star, you'll get 100% in your exam, okay, in all your exams, okay? Right, got a bit of a treat for you. Oh, this is very cold. This has been in the freezer. Okay, and it's sealed. And the reason why it's sealed is because, look at that biohazard specimen bag. And the reason why it's sealed is because in this jar, we have something very special. I'm going to show this to you on the camera. That's some things that are very special. Okay. Now, I'm going to try and make sure that you can actually see inside. Okay. But you can actually see some grains. Let's see if have you got my phone? Okay, bring the light really close. That's it. Okay, right. Bring it from this angle here. There we go. So we've got an assistant. There we go. Okay, so blow the screen up. Can you see some gold, yellow balls and some blue crystal balls in there? Okay, can you guess what that is? Boomy, you're absolutely correct. You can switch the torch off. These are genuine gallbladder stones. These are gallbladder stones, okay? They're crystallized cholesterol. And your gallbladder is about the size of your fingernail. So you can already see that there's a lot of this stuff that would have had to have fitted in here, okay? And these stones developed over many, many years. There are, I would say, about 200. But I'm never ever good at guessing these. This is what they managed to get out, okay? Now, when they do the surgery, it's all done inside. It's all done inside. So you have like 2 rods that go into your body. What happened is they have to cut it on the inside, and as they touched it, I was told that it just sprinkled out because the membrane on the gallbladder was so thin because of the pressure. It burst open, so that's all they could actually scoop out at the time and catch. And there were like lots of little stones that were washing around my body. So yeah, that was quite an interesting one. That took me out for about 4 weeks. So that was linked to bile. Okay. So pancreatic juice, another fluid, flows into the duodenum. It's called bile. So that bile is constantly now flowing into my digestive system. But that bile, here it is. That's your liver, okay? But that bile used to be kept in a little pocket over here, tiny pocket called a gallbladder, okay? And now that pocket is no longer there. That little satchel is not there, okay? So the bile now flows directly in from the bile duct into that gallbladder was there, okay? You can see that little bag type structure, and they had to remove that. And if the wrong type of cholesterol forms in there, is produced in there as a result of being over 40, being a little bit rotund, shall we say, a little bit obese, a bit fat, overweight, I should say. And also your diet, so you've got to watch your diet. Then what happens is your body produces a lot of different type of cholesterol, so I was told, and then that starts to crystallize and it just gathers there. The bile actually contains yellowish bile pigments as well. So the bile breaks down the red blood cells as well. Now this is what you've been reading about. So yeah. And it doesn't contain enzymes. The bile. What the bile does is it's just a fluid that goes up to a big fat globule. I'm not being offensive to the fat globule. It is actually literally a fat globule. And what it does is it breaks it down into smaller pieces, and then that increases the surface area. And I think we went through this exercise where I asked you to get a piece of paper and to color it on the outside and then open it up, and then we talked about surface area. So it allows enzymes later on to attack the surfaces, whereas before it would have taken a bit of time to digest. Okay, so breaking it up means that you increase the surface area for the enzymes to act on. Cholera? Did you understand cholera? When you read about cholera, did you understand it? So the cholera bacteria are ingested and multiply, so they multiply in your guts, then the bacteria are attached to the wall of the alimentary canal. That's where they release their toxin. And then you have chloride ions to be released, and then the release of the ions cause water to move into the lumen by osmosis, which is very clever. And then you have a lot of water in the canal, and that means that you have watery diarrhea. So the water actually goes from your bloodstream into your alimentary canal, into your guts, and that's what makes it watery. Do we have to study the treatment? Yes, you do have to study the treatment. The treatment is in the page. Can you see it in the page? Yes, cholera treatment in Haiti. When fluid losses are very great, rehydration therapy can be given. So all of that is in the supplementary section. That's why you've got the big brown line going through. So the answer is yes, you do have to study the treatment. Okay. But isn't it a case of, as long as enough fluids can be given to replace the losses of chloride ions and the losses of water, your person will recover. It's very interesting to see how the bacteria release the toxin, and the toxin causes the chloride ions to be released from the walls of the alimentary canal. And then as you've got an increase in ion concentration, you got osmosis that happens backwards. So very, very interesting. The nutrients dissolve in the blood plasma and then taken to other parts of the body where they become assimilated as part of the cell. So everything gets ferried off to the liver. The liver breaks down some of the nutrients. It converts it to other substances. Some of the nutrients are stored, and then the remainder are left unchanged, and then it gets funneled back off to other parts of the body, and then it's what's called assimilated. Make sure you know about glycogen as well. The liver has an especially important role in the metabolism of glucose. If there's more glucose than necessary, the liver will convert some of it into the polysaccharide glycogen and store it. Okay?
Sir,
Yeah.
I was asking to explain the information in our exam. Like, do we have to do the same thing as in the PowerPoint? Or just draw it and name the, like, do we have to also explain, like, what is chloroplast and nucleus in this exam?
What do you think? First of all, I'm going to ask you, what do you think?
No.
Okay. So you think that you don't have to answer it, okay?
Yeah.
All right. Okay. So the best thing to do for this one, I'm going to show you something. GCSE Biology, IGCSE, Cambridge. Okay. Describe and compare the structure of a plant cell with an animal cell. Okay. So identify things like mitochondria. State the functions of the structure seen under the light microscope in the plant cell. That's your answer. Okay. That's your answer. So you were asking, is it just the names, or do we need to know the functions? And that answers your question. Okay, you need to be able to state the functions. You're welcome. You're welcome. And hopefully, by taking you through how to find it, you are now a better learner, or better equipped learner, because you are now able to, when you're studying your GCSE, it's something that you weren't really doing when you were in Key Stage 3, as a Year 8 student. We didn't say, oh, go to the specification, go to the syllabus, go find out. Now you'll find that every GCSE teacher will be encouraging you to look at the specification. Why? Because they want you to be able to use the time that you've got to the best effect. So if it's a great question. You're looking at it and you think, is it just extra detail? Is it just to make the slide look pretty? Is it just to make it interesting? No, it's actually a deeper level. Yeah, you do need to know it. So I'll keep that open, but thank you for your question. Cool.
You're welcome.
Yeah, good. How are you? Are you staying safe at the moment?
Yeah.
Good. Give my regards to your family. I hope they stay safe too. Catch you soon. Okay. Do we have to draw the plant cell structure? Okay, so do we have to draw the plant cell structure to identify the different levels of organizations in drawings? Yes, you do need to. Okay, so the strict answer for your question, Bhumi, is no, but I would expect a person working at the top of GCSE to be able to do that as a skill. Does that make sense? Cool. Okay. All right. Absorbent spectrophotosynthetic. You need help with this. Okay. Right. So, let's have a look. So, Sultan, come back on the microphone. Okay. Right. So, how can I help with this one? So, which parts look really?
I don't understand
like any of it.
I don't understand.
All right. Okay. So, light is made of a number of wavelengths. White light is made of different colors.
Yeah.
Okay, right. So each of those colors has a property. And it all depends on how, because I know you don't do physics, do you?
I do physics.
Oh, you do physics, right. Are you familiar with the electromagnetic spectrum?
Kind of.
Kind of, right. Okay, so the electromagnetic spectrum is a way of arranging all of the different types of electromagnetic waves that can have different wavelengths. I'm going to do a little bit of drawing here. This is going to entertain you. Okay, so if I...
Okay.
Yeah, I know. It says going to start doing drawings. Just hold on a second. So if we have red light, oh, that's not red. Let's have it. Red light looks like this, okay? Keeping a very steady hand. All right, so it's got a very low wavelength, and the wavelength of light, of any wave actually, is this distance here. I'll show you. Okay, it's the distance between this point here, okay, and where the wave hits that position again. Okay, that's called a wavelength. All right. So that is, oh, that's far too big, isn't it? Let's go here. Wavelength. Okay, and we actually use a very small distance to use this, to measure this, called nanometers, okay? All right, so that's the wavelength, all right? And for blue light and things on the blue end of the spectrum, the wavelength is much shorter. I'm going to have fun with this. And these are more energetic waves, okay? There we go. All right. Do you get the picture? All right. Okay, so the wavelength of different colored lights, if I just do that, go right across here, you can actually see that the wavelength is a much shorter wavelength. Do you get that? Is he still there?
Yes, I did get that.
You get that? You're getting me, yes? You're getting me?
Yeah.
Okay,
Yep.
so, good. You're getting me. Right, so if I now switch that picture, so keep that in mind, okay? And now what I'm going to do is I'm going to switch over. So the shorter the wavelength, the more energetic, and the more towards the blue end that is, right? And now I'm going to switch over back to the presentation. So everything on the blue end. So you know your colours of the rainbow, don't you? Okay. So everything on the blue end should have a smaller wavelength. Okay. Shorter wavelength. So if we zoom in, oh, where'd it go? There it is. If we, I can't zoom in on that one. So if we have a look, I'll just read off the numbers here. On the left-hand side, where it's blue, it says 400. Yeah? Are you with me, Sultan?
Yes.
Okay, so on the left-hand side, it's got 400, and that's blue, correct?
Mm-hmm.
Okay, and on the right-hand side, it's more on the red end, and it's 700. That means that the wavelengths of red light are longer, almost double, okay? All right, so this is basically, in a nutshell, telling you what the different wavelengths of light are that are in light. This would be in sunlight, okay? So there's blue color, there's green, there's yellow, there's orange, there's red. That's how we can see rainbows, right? Now, so that's the x-axis, all right? Now, the y-axis, the one going up and down, is telling you about the amount of light absorbed, okay? So we can see, and this is all to do with photosynthetic pigments. This is basically the pigments that are inside plant leaves, all right?
Okay.
And chlorophyll is a pigment that will absorb certain parts of light, certain wavelengths of light. Now, the text says chlorophyll is green, so it reflects green light and rates of photosynthesis is low. Right, so plant leaves are green because the white light is made up of red, orange, yellow, green, blue, indigo, and violet. And that is what you can see at the bottom. Yeah, you can see those colors. And you can see where it's yellow and green, these graphs are very low compared to the blues and the dark blues and the purples on the left-hand side, and compared to where these graphs are very high on the red and the orange parts. Can you see that? So you see the peaks are towards the blues and the purples, and there's also big peaks on the red and the orange. Yeah? And it just goes completely flat. So it's like two sets of mountains on either side. Yeah, two jebels, right? And then you've got a big flat plateau right in the middle where it's almost zero, correct?
Yes.
All right, okay, good. So the higher the peak, the more that particular color is being absorbed by a pigment in the leaf. Okay, so the graph is showing you what's actually happening to white light as it shines on a leaf. And the higher the peak, the more that color is being absorbed by that particular pigment. So there are three pigments that are in this graph. There's chlorophyll A, chlorophyll B, and there's a set of chemicals called carotenoids, okay? And carotenoids, interestingly, are orange in color. They give the orange color to carrots. So you can think of it like that. Okay, chlorophyll A and chlorophyll B are two different types of chlorophyll. So you can actually see there's lots and lots of peaks because there's lots of, on the left-hand side, there's lots of absorbance of blue light, purple light on the left-hand side by all three of these pigments, okay? On the right-hand side, you can see that the chlorophyll also absorbs the orange and the red end. So none of these pigments seem to be touching the yellows and the greens. So any light that falls on a leaf is going to have absorbance. So if you think about it, now you can look at the picture where I'm actually describing what's going on. So you've got red light, orange, green, blue, indigo, violet. All of those light wavelengths fall onto a leaf. If the leaf is not absorbing it, that wavelength of light will be reflected. So leaves absorb things right on the end of the blue end and right on the end of the red end, and everything else which is in the middle, which are the greens and the yellows, are reflected back off a leaf because these pigments don't absorb those wavelengths of light. Okay, and that is the reason why leaves look green. And the reason why these pigments are absorbing light is because of photosynthesis. These are the right wavelengths of light that trigger off the whole process of photosynthesis. And that's it. If you do have questions, it's really essential that you do ask questions through the DMs, through the personal messages. You'll find that this week's work is easier to access. In actual fact, you'll possibly find that it's easier to access across the board. We've responded to some feedback and we've placed some video guidance on each of the lessons. So hopefully it will become a lot easier. And that's about it. So I shall bid you goodbye. You can flick your microphones on and have a bit of fun. Just say goodbye to each other. Bye. I think that was Abhishek. I can recognize your voice anywhere.
Goodbye.
Yeah, bye.
Have a nice day, sir.
Bye-bye.
Have a nice evening, sir.
Yeah, I'll have a nice afternoon and evening, Sultan. Thank you.
سالم أحبك.
Thank you. All right. Take care.
Thanks for the help, sir.
Oh, no problem. It's what we do. It's what we do. Try and remember you're not alone. Hello,
Have a good day,
Simon.
sir. Hi,
You
sir.
too.
How are you?
You take care. You take care. Good to see you on. Good to see you on. Are we going to say hello to Mohammed as well? Is Mohammed Khanzada?
حي سلطان شحالك؟
Good. All right. Take care, you guys.
Okay.
Take care, sir.
Okay. See you soon. Take care. Stay safe. Yeah.